pleasure to introduce the next lecturer, who is Surya Ganguli. And Surya, his background is really pretty much everything. He studied electrical engineering, computer science, physics, and mathematics. Then he did his thesis in string theory. And then, well, I never asked the story, but for some reason his postdoc was in neuroscience. And then he jumped into neuroscience. And you know, with all that breadth, he ended up, you know, at the moment he is in Stanford and covering this intriguing mix between statistical mechanics, neuroscience, and theory of machine learning. And I think that's what he will try to tell you about in his lecture. So it's a great pleasure to have you here.
Okay, thanks, Lenka. Can you guys can hear me? Okay, great. Yeah, so yeah, I think that there's a really kind of intriguing field at the intersection of neuroscience, machine learning, and statistical mechanics, and more generally theoretical physics, applied math. And I'll kind of try to explain why. So of course, there's a lot of excitement in neuroscience, and why is that? Partially because of technological breakthroughs that allow us to witness the immense complexity of neural systems across multiple scales of organization, from molecules to behavior. So we can do all sorts of recordings of the brain and even stimulation of the brain and record the response. So we can really peer within the brain in ways that we really couldn't do before. So in order to understand this complexity, it's actually useful to turn to fields that you're, of course, familiar with, and the physical and mathematical sciences that themselves develop powerful tools to deal with complexity and try to confront it. But those tools aren't enough. We need to develop them further. But of course, another really intriguing aspect of things is that neural systems aren't simply tangled webs of complexity that exist for their own sake, like gels or colloids or things like that. They're actually been evolved. They have a purpose. They have a function. They've evolved to solve important computational problems. So it's actually really interesting to turn to ideas from the engineering sciences, especially machine learning, information theory, control theory, and so on, for inspiration as to the algorithms that the brain is implicitly using to solve computational problems, and indeed to elucidate what actually are the computational problems that the brain is solving in the first place. So it's an incredibly interdisciplinary subject that uses all of these ideas. And you know, on a day-to-day basis, I interact a lot with experimental neuroscientists at Stanford and elsewhere. And we work on a variety of problems, like how the retina sees, how flies detect motion. This is a pretty cool one, how mice navigate. I don't know, how many of you guys have heard of grid cells, by any chance? Wow, surprisingly, relatively small number. So these are these amazing cells that fire in the mouse's brain, in the medial entorhinal cortex, an area related to navigation. They fire if and only if the mouse is at the vertices of a hexagonal grid in this space. So what you're looking at, the black are the trajectories of a mouse moving in a square arena. The red are the points where the cell fires. Okay? So amazing kind of, it almost looks like Turing pattern formation, but inside the brain. We've also been looking at how monkeys reach, how genetically modified circuits can become smarter and learn faster, and so on. But if we kind of zoom out a little bit and try to think about sort of the big picture of where this field is going, I think there's kind of two major issues that we kind of have to confront. Okay, so on the one hand, we have this palpable technology-driven excitement in neuroscience, various methods for measuring the dynamics of many, many neurons in the brain, various methods for measuring the connectivity between these neurons, various methods for perturbing the circuit and then recording the response. But the question is, how do we go from this explosion of data to a conceptual understanding? There's kind of two issues, right? Issue one is that we will not soon be able to record from the entire brain at single-cell resolution during any complicated behavior, okay? Either in a mouse or a monkey or a human. Okay, but let's say even if we could, let's say we could record from all the neurons in the brain during any complicated behavior, what would we do with that data? How would we confront such a complicated large data set? Okay? And I think some ideas, just again, zooming out, just kind of general theoretical directions that can help us in dealing with these issues, is one, high-dimensional statistics. Basically, how can we understand neural circuits at such a subsampled measurement regime where we're recording from an infinitesimal fraction of the total number of neurons in the circuit? And second, even if we had all the data, what would we do with it? Well, we could look to our colleagues in computer science for interesting challenges. Our colleagues are generating interesting neural networks that do interesting things, and we could look to those networks as sort of a quote-unquote warm-up problem. It's a very challenging problem in and of itself. And so deep learning, you know, which is also gathering a lot of excitement, is kind of becoming relevant for neuroscience in multiple ways, which I'll explain. Okay, so I just wanted to kind of give an overview before we dive into these things. So high-dimensional statistics, why might it help? Well, the basic idea is that you've seen examples of this, but at the core of every data analysis algorithm, there lies essentially an implied hypothesis about simplicity in data. Okay, so basically, if you believe that your data lies in a low-dimensional manifold and a high-dimensional ambient space, and we'll see examples of that, then dimensionality reduction is a very good algorithm to use to look for a simpler description of your data. If you believe there are independent sources that are mixed that contribute to your signal, then independent components analysis is a good algorithm. If you believe there are clusters, there's various clustering algorithms. In graphical models, if you believe there are no strong loops, then message passing or belief propagation, which you've seen, is a good way to compute marginals. If you believe the signals that you're looking for are sparse, then compressed sensing is a good way to recover them and measure them. And also, if you believe that you're searching for matrices that have low-rank structure, something called nuclear norm minimization is a great way to find them or denoise such matrices. But the basic take-home message is if any of these simplicities exist in a system, then we can often accurately characterize the structure and function of that system using many fewer measurements than the total dimensionality of the system. In its relevance to neuroscience, the total dimensionality of the system is the number of neurons in the brain. In the human brain, it's about 10 to the 11th. We're never going to record all of them. But in certain circuits, you know, even in certain restricted circuits solving a task, there might be 10 to the 9th neurons. We won't record from all of them anytime soon, and you'll see why. But you can still record 100 of them and actually get a pretty accurate picture of the underlying algorithm that circuit is exploring. And that's essentially because of the low dimensionality of neural recordings, we'll see. So we're going to kind of focus on dimensionality reduction. Okay, now what about this second issue, examples of complete neural circuit models and deep learning? Why might this be relevant? So I want to motivate kind of a new type of alliance between neuroscience and machine learning that will help both fields. And this is laid out in an opinion piece that we wrote recently, not so recently now. But the basic idea is the following. You know, in theoretical neuroscience, we're kind of in it because we want to understand how the brain works, okay? But that's a very fuzzy question. What does that really mean? What are we actually trying to do in this field? Well, a more proximal version of that question, a more concrete version of that question is, if we can understand how the connectivity and dynamics of a neural circuit gives rise to behavior, okay? And of course, learning is important, right? The connectivity in your brain is sculpted by experience. We'll see simple examples of that soon. And so we'd also like to understand how neural activity and synaptic learning rules conspire to self-organize useful connectivity that subserves behavior, right? So, oh, I took it out. I had a dig on physicists, which I unfortunately took out. But it's actually very useful. Let me put it back in. There's an intermediate bullet point that I don't know why it's not here. It's not enough to study random networks, okay? There's been a long history of physicists analyzing the dynamics of randomly connected neural networks. Actually, it was very useful. Like, we understood a lot about the phase diagram of these networks. But again, such random networks are not mimicking what's going on in the brain, because what's going on in the brain is that the networks were evolved or learned to solve a particular computational problem. Okay? So again, it's not enough to study random networks. But the field of machine learning has generated many, many neural networks that accomplish lots of interesting functions. Machine translation, you know, transferring images to text, a text-based description of images, and so on. These are functions that we know of no other way of doing in any artificial system at the moment. Okay? What's a little bit scary is that we know everything about these networks. Any experiment we want to do, we can do on these networks and get the answer. We know their entire connectivity. We know the dynamics of every single neuron. We know the learning rule that was used to train the network. We know the entire developmental experience of the network in terms of its exposure to training data. Yet we don't have a meaningful understanding of how they learn and work, nor do we even have a benchmark for what that understanding might even look like. Okay? You've heard about this brain initiative where a lot of money is being poured across multiple countries, not just in the US, in order to get large-scale measurements of neural activity, get the connectivity between the same neurons, and so on and so forth. It's a little bit scary that we don't even know what we would do with that even if we had it, right? Even in an artificial system. I don't think for a second that if we understand these artificial systems, that that understanding will directly transfer to the brain. But the methods that we use to arrive at that understanding might be quite interesting and might sharpen the questions we ask in neuroscience and the way we design experiments in neuroscience. Yeah?
Do you think that means that it's too early to invest in a large-scale problem like the brain initiatives?
No, I don't think so. I think as long as the scientific questions are targeted to restricted behaviors that we can dissect. Well, I'll talk about that soon, right? So I mean, these initiatives that will last about 10 years won't solve the question of how the brain works. But they might shed insight into more limited situations, like how do we adjudicate between multiple options? How do we maximize a reward? How do we resolve ambiguous stimuli? There's a whole host of questions that are incredibly intriguing. And I'm super excited about this data. But we're kind of, what we'd really like to do is we'd like to put theory in the driver's seat in neuroscience so that theory drives the design of experiments. That's been happening a lot in physics. It's, you know, the interaction between theory and experiment is much less mature in neuroscience. And there, I'll give you examples of how we can do that here. Yeah? We're going to discuss both. So I'm going to start with models of single neurons soon as we start diving into the real material. I'll explain what I mean by that. Okay, and believe it or not, a lot of experimental neuroscientists actually buy this argument. They think, yes, we really need to think about what it is we're doing, and our friends in machine learning can help us. Okay, so this is an outline of the talks. There's a lot of stuff here, and I probably won't be able to cover all of it, but you'll have all my slides, you'll have all my notes, and so on. So I wanted to start with the kind of classic stuff that's very old, but they're oldies but goodies, right? They're really like, you know, I'd be ashamed of myself if I gave a lecture in neuroscience and I didn't discuss this stuff. But I'm not going to go into it in super detail because it's very old stuff. There's lots of review articles on it. I'll point you to references in the original literature. But I just want to take a quick quote just to get a level of sort of knowledge in the audience. How many of you have studied the Hodgkin Huxley model of a neuron? Okay. Okay. How many of you have studied the Hopfield model, maybe simulated it? Okay. So it's usual 50-50. The perceptron. How many of you know principal components analysis? You should all know that by now. Okay. So it's 50-50. So I'll have to go a little bit more slowly than I thought. But that's fine. Then the second lecture, this will roughly be divided in 3 lectures, so there may be spillover. The second lecture, we're going to discuss high-dimensional statistics, like what is it that we can do or correctly recover about the algorithms a neural circuit uses to accomplish this task without recording all the neurons in the circuit. And this will actually, you know, we'll discuss, for example, what's the best way to do regression in high dimensions, and this will actually use replica theory. We'll discover how to accurately recover neural state space dynamics, and this will actually employ random projections, random matrices, free probability. Some very beautiful mathematics will be involved. And actually, I put this in because of Florent's lecture. While Florent was talking about tensor decompositions, at the same time I was editing a paper that we're writing on tensor decompositions, but applying it to neural data to figure out how neural circuits learn by decomposing tensors. So this is kind of hot off the press. And then in the third lecture, we'll move on to deep learning theory and practice, where I'll discuss a sequence of papers where we actually used ideas from physics to come up with better understanding and learning algorithms in deep learning theory, and also applying deep learning to the brain to understand the brain. So yeah, we'll discuss that. There's a review article that I wrote with Madhu and Shubhi, a student and a postdoc in my lab, that covers actually a lot of the basics of statistical mechanics of complex neural systems and high-dimensional data using replica theory. So in a unified way, we discuss spin glass models of neural networks like the SK model and the Hopfield model, the statistical mechanics of learning, the perceptron, random matrix theory, random projections, compressed sensing, and future directions. And we do it all in a very unified way using replica theory. So if you're interested in this subject, you can see how replica theory in a unified way can get you all of these results across all of these disparate fields. And so we've kind of put that up on the website. Okay, so let's get started. So just so that we're all on the same page, I'm going to start. Two of my most favorite models in neuroscience are actually the Hodgkin Huxley model and the Hopfield model for very different reasons. The Hodgkin Huxley model won a Nobel Prize. It's probably the best, most quantitative model we have in neuroscience that predicts in detail the shape of an action potential, what makes a neuron fire, the basic unit of information in the brain. We'll discuss that soon. The Hopfield model is extremely different. It doesn't make any quantitative prediction whatsoever about how neural activity patterns should behave in the brain, but it provided at the time a very conceptual overview of how memory might work in our brain in a way where the concepts weren't really there. So let's start off with Hodgkin Huxley and just what is a neuron, so that we're all on the same page. And we'll start off with some real biophysics, but we'll quickly let's see. We'll quickly ditch the biophysics so that we can look at collective properties. So let me draw in caricature a neuron. Okay, it has multiple branches coming into it. Huh?
There's a light switch on the side here.
Oh, there's a light switch on the side. Oh, perfect. Oh, excellent. Okay. Okay, and these dendrites, so these incoming connections, these dendrites, come into a soma, and then the soma, which is the cell body, sends out an axon, and then this axon branches and goes to other locations. Okay? So, and then other neurons, the axon of another neuron, can form a synapse onto this neuron. This is called the postsynaptic neuron. If we think about this as the synapse or the connection between two neurons, this is the postsynaptic neuron, this is the presynaptic neuron. Okay. All of the information that's signaled by a neuron is contained in the membrane voltage across the membrane. So if we zoom in on this segment of the membrane, okay, the membrane is a lipid bilayer. So there's hydrophobic components pointing in. So this is a lipid bilayer. And inside the lipid bilayer, okay, so actually what happens is, because of non-equilibrium processes, it turns out that there's an excess of positive charge on the outside relative to the inside, right? So there's a weak electric field going down. So there's a membrane voltage, which by convention is negative, okay? So there's a resting membrane voltage, which is less than 0, okay, because of this asymmetric charge distribution. Now, all of the information processing in the neuron is transmitted by this membrane voltage, by excursions in this membrane voltage. Now, that's not all there is in the membrane. There are these voltage-gated ion channels, okay? The most important one is the sodium channel. So it turns out that sodium has a higher concentration outside the cell than inside the cell, okay? But at this resting voltage, these sodium channels are closed, okay? Now, what happens here is if, for any reason, the membrane voltage increases, okay, then the sodium channels, this chalk is, the sodium channels open. big but less bulky chalk. Okay, it's all pretty bulky. Oh, excellent. Okay. Perfect. Hopefully you don't mind yellow. There should be white as well. Okay. Yeah. Which box? This? Oh, here. Okay, got it. Okay, perfect. That's all. It's premium ivory, but it's yellow. Okay, well, let's try this. So, so what happens is if the voltage increases, then these sodium channels open. But what happens if sodium channel opens? If the sodium channels open, because there's a chemical gradient of a concentration gradient of sodium going down this way, sodium will rush in. Okay. So then much better. So then Na plus rushes in, right? But then what happens when Na plus rushes in? How does the membrane voltage change? It increases, right? Because plus charges are rushing in. That reduces the voltage drop. So then that creates a positive feedback loop. Now, the sodium channel is fast, so it opens very fast. So basically, this is a fast positive feedback loop. Okay, but that's not the only ion channel at play in the neuron. There's also a potassium channel. Okay, and potassium has an excess concentration on the inside of the cell. Okay, and this is also a voltage-gated ion channel. At rest, this ion channel is closed, but it turns out that again, if V increases, then what happens is the sodium channels, the potassium channels open, but they open slowly. Okay, and then what happens is K plus is going to rush out. Okay? So now what happens if K plus rushes out? What does that do to the membrane voltage? It goes down, right? Because positive ions are going out, and that increases the electric field this way, and the membrane voltage drops according to the sign convention that I chose. So this inhibits further increases in the membrane voltage. Okay? So this is a slow negative feedback loop. Okay? So this kind of meme of fast positive feedback and slow negative feedback is a meme that appears all across biology, and that's the essential idea that gives rise to an excitable system, right? So if there's a voltage deflection that goes above the threshold for sodium to open, you get this positive feedback effect. And so if this is time and this is voltage, you're at some resting state voltage, which is typically about minus 60 millivolts. That's the order of magnitude. And if there's a small increase in the membrane voltage and the sodium channels don't open, the membrane voltage comes back down. But if there's a big increase in the membrane voltage above the threshold where sodium channels open, you get this huge deflection. Then the potassium channels open, and there's a little bit of resetting dynamics. So here Na plus is going in, and here K plus is going out to bring the voltage back down. And this is the anatomy of the spike. Okay? This whole process unfolds very rapidly in about 2 milliseconds. Okay? Just to give you a sense of the time of cognition, if you were to show a person images of, say, an animal versus no animal, and the person has to decide, it takes you about 150 milliseconds before you see signals in your brain that reliably detect animal from no animal in an image. So there's kind of multiple action potentials that can occur that can give rise to these cognitive things. Yeah.
Does it mean the sodium stops going out?
Yeah, there's something that I glossed over, is that the sodium has an inactivation gate, actually. And so at high voltages, it inactivates as well. Okay? So I glossed over that. So it turns out you can write down a set of different ordinary differential equations, a four-dimensional dynamical system, where the degrees of freedom are the membrane voltage, the level of sodium activation, the level of sodium inactivation, and the level of potassium activation of these ion channels. That gives you four variables. You can write down a couple dynamical system. You can solve it numerically, and you can get the shape of the action potential. You can compare this to recordings. Hodgkin and Huxley did this from the squidge giant axon, which was this huge axon that you could actually see. And they got a quantitative match between the model and the actual experimental recordings.
Just a silly question. How does the sodium get big out?
Excellent question. Yeah, so you have to pay a price, right? There's a constant energetic cost for being excitable, which is you have to reset the system. So you have these extra pumps called the sodium-potassium pump that basically you can think of this as like a flywheel that continuously pumps sodium out and potassium in. Okay? And it burns ATP in order to do that, right? So basically, you're burning ATP, you're spending energy to create a non-equilibrium steady state. And because you now have a non-equilibrium steady state, you can do rapid signaling in a way that you couldn't do if you started from equilibrium. Right? So this is the idea that life is fundamentally at non-equilibrium, and equilibrium is like tantamount to death. Right? No, I'm literally true. I mean, if you've equilibrated, you're in big trouble. All right? Yeah.
I'm still a little unclear. So there's a potential difference between the outside and inside the cell wall?
Yeah, exactly. Yeah, it's a cell membrane.
Cell membrane.
Yeah.
And then this schematic right here with the sodium, where is that located?
Okay, so yeah, these sodium channels are embedded within the cell membrane. Right.
Okay.
So these are basically very complex macromolecules that deform their shape in the presence or absence of electric fields. In and out of the, yeah, exactly. So you can roughly think of this thing has like a little blocking part of the macromolecule. And if the electric field is strong enough, then it's blocked. But if the electric field weakens, it can move out. And that allows sodium to rush in.
Well, what is the relation of that to the dendrite coming from the other?
Okay, yeah, we're getting there, yeah. This is all about what's going on at the cell membrane. And I've only been talking at about a single point. There's also the passive propagation of membrane voltage, because you have this system replicated everywhere, and there's kind of this diffusive coupling. So if the membrane voltage goes up here, that will create a propagating wave that propagates downstream and can excite downstream neurons, okay? But you're asking essentially the most important question is, I said if the membrane voltage increases, right? What could cause the membrane voltage to increase high enough that sodium channels open and you get this spike? Okay, so of course, this reminds you of explanations for God. Like, if you can't explain something, that you have to blame it on something else. So we're going to blame it on the presynaptic neuron. Okay, something caused the presynaptic neuron to spike. All right. So then the presynaptic neuron, the spike, this membrane voltage deflection, comes to an end at this cleft. This is called the synaptic cleft. It's a 20 nanometer kind of width. By the way, we're going to forget all of this, okay? But I just wanted to get you grounded in real biophysics first before we start making simplifications, so that we can look at collective effects of multiple neurons, which is what we're all sort of interested in. Okay, so what happens here? There's a complicated process. So let's imagine a presynaptic spike comes in, right? So you have the presynaptic spike. That's a voltage deflection. Okay. What does that do? At the end of the synapse, there are these voltage-gated calcium channels. And because the voltage increases, the calcium channels open. So that causes calcium to rush in. Now, calcium is a ubiquitous kind of signaling, small signaling molecule in cells that does all sorts of things, but at the synapse, what it does primarily is it opens vesicles. Vesicles that contain neurotransmitters. Okay? So let me zoom in on the synaptic cleft here. So here's the presynaptic neuron. Here's the postsynaptic neuron. Here's a gap. There are these little vesicles just waiting that contain neurotransmitters, small molecules like GABA or glutamate. And when calcium rushes in, that triggers machinery for the vesicles to fuse with the membrane and release the neurotransmitters into the extracellular space. They diffuse. Now, diffusion is, of course, slow transport, very slow, right? So there must be a very good reason that the brain does this, and it turns out there is. But fortunately, it's not so slow because the synaptic cleft is only 20 nanometers wide. But these transmitters diffuse across the synaptic cleft, and then what they do is they open in the postsynaptic neuron they open neurotransmitter-gated ion channels. So, voltage can open an ion channel, but also chemicals can open an ion channel, right? They open these neurotransmitter-gated ion channels. Okay? Now, as soon as an ion channel is opened, ions can rush in or out of the cell. Okay? And so depending on the type of ion channel that's opened, you could either increase the membrane voltage of the postsynaptic cell or decrease it. If you increase it, that's an excitatory synapse, a positive Wij, as we'll see soon. If you decrease, it's an inhibitory synapse, a negative Wij. Okay? And that's basically it. Now, why did nature go to all of this complexity to convert an electrical signal to a chemical signal back to an electrical signal? It's because this provides a site for the modulation of synaptic strength. There's many, many biophysical degrees of freedom that can govern the gain from a presynaptic spike here to the magnitude of the postsynaptic deflection here. For example, you can modulate the number of vesicles that are released. You can modulate the number of postsynaptic neurotransmitter-gated ion channels that are released. By modulating all of that, you can change the strength of a synapse. And these are long-lived chemical degrees of freedom, so they can contain memories that last a lifetime, right? So actually, all of your memories are stored in this molecular, we think all of your memories are stored in this molecular machinery that governs a strength of connectivity between neurons in your brain, which ultimately determine the flow of electrical patterns of activity through your brain. Okay, so if we have to model all of this stuff, we're toast, right? We're never going to be able to understand the entire brain if we have to model it. But fortunately, we're used to the idea of coarse graining. And there are reasonable ways to coarse grain the system, where each time you coarse grain, you lose a little something, but you're willing to give it up. So there's a very nice paper called Model Neurons from Hodgkin Huxley to Hopfield that kind of goes through this coarse graining, starting from Hodgkin Huxley and going to a simple spin that we're all used to. Oh, yeah, so Hodgkin Huxley is the entire machinery that describes everything on this board, right? It's the set of 4 differential equations where one of the variables, sorry, it's a set of differential equations over 4 variables where one of the variables is the membrane voltage, okay? The other variables are associated with the degree of activation or inactivation of the ion channels, which are an important part of the state of a single patch of the membrane voltage, yeah?
How big is noise and fluctuations in voltage? Can you estimate?
Yeah, that's an excellent question. So these ion channels, of course, are stochastic. You can model them as Markov chains. And so they have kind of Bernoulli-type fluctuations. But you have a relatively large number of ion channels, but not too large, in any patch of membrane, So you get a 1/√n type of reduction in the fluctuations, where n is order 10 to 20 in a relatively small patch. So the fluctuations aren't as large as you might think, and neurons are capable of precise timing. if needed to. Okay. All right. So okay. So you know, one thing about these spikes is that there's no information carried in the shape of the spike. It's an extremely stereotypical pattern that's basically whose dynamics is determined by the dynamics of these molecular complexes, the sodium channels and the ion channels and their gating kinetics and so forth. So there's really no information in the spike. And again, like I mentioned, cognition happens on the time scale of hundreds of milliseconds. The spike is 2 milliseconds. So we can exploit timescale separation to come up with simpler models that don't model the dynamics that gives rise to the spike and just treat the spike as a punctate event. So this is this leads to the workhorse. If you care about the timing of spikes, this leads to the integrate and fire neuron. Which models only the membrane voltage, and it basically gets rid of all of the other degrees of freedom because they're faster relative to the timescales that we're actually interested in. So we can think of the integrate and fire neuron as just a leaky integrator of membrane voltage, right? So this membrane voltage also has a passive decay to the resting membrane voltage in the absence of the ion channels. So this is the passive decay. And then there might be external currents that come in. These external currents could be coming from a presynaptic neuron. You can inject them in if you want. And then what happens is you have this additional rule for spiking. If the voltage ever becomes bigger than V threshold, then you emit a spike. And then you set the voltage to V reset. Okay? So what does the dynamics of this neuron look like? Let's say this is the voltage axis, and this is the time axis. The way I've set it up is I've just set it up so that V rest, like if in the absence of inputs, the voltage will go to 0. So V rest is at 0 here. Let's say V threshold is up here, and let's say V reset is down here. Okay? So let's say that the input is weak, the external current is weak, so that I is less than V threshold. Okay? So then what happens is the membrane voltage it's just a leaky integrator, so it will integrate the input, and let's say it's just a constant input for now. It'll integrate the input, and then the membrane voltage will stay below threshold, and the cell just will not fire. Okay? However, if the input current is above threshold, so you've noticed that I'm playing fast and loose with units. There's some capacitances and so on that I'm just ignoring. But in any case, if the input current is bigger than V threshold, right, then what will happen is it will integrate to a level that's above threshold, so it'll hit threshold, then it'll emit a spike, the membrane voltage will be set to V reset, so this yields a small refractory period, then it'll integrate again and spike, then it'll integrate again and spike, and so on. So basically, in response to a constant input that's above threshold, it spits out a constant rate of spiking. Okay?
This is the constant input from the previous synapse?
Yeah, okay, so inputs from previous synapses are never constant because they're punctuate events. So this is, so we can think of this as like an experiment where the experimentalist is just injecting a constant current in. These are actually experiments that happened a lot early on. Experimentalists just wanted to see what happens if I just inject a constant current into my cell. Okay.
But in the physical system, it's spikes that are coming in.
Exactly, it's spikes that are coming in, and I'll tell you how to model that soon. Okay? Next, actually. Okay, so actually, that's exactly what I was going to do next. Do I have space here? Yeah, okay, so this is how the cell responds to a constant current, but how do I have a model network of integrate and fire cells? So basically, I need a model for what the synapse is doing. Like, let's say the presynaptic cell spikes, that's going to effectively inject some current into the postsynaptic cell by opening these neurotransmitter-gated ion channels, right? So I need a model for how a presynaptic spike injects current into the postsynaptic cell. And so that you can basically do that through the following equation. The current coming into neuron I at time t is a, so let me put this up here, is a sum over all the other neurons in the network times some synaptic weight, Wij. This summarizes all of this molecular machinery that governs the gain from a presynaptic spike to the postsynaptic response. And then I sum over all the times of the presynaptic spike, right? And I have a little canonical function, a kernel function, that's just t minus tj. And this function, delta t versus k, is just like a little alpha function. You know, it rises and then falls. So basically, each time a spike comes in from the presynaptic neuron, you inject a current into the postsynaptic neuron that has a particular time course. Okay, and it's gain modulated by the synaptic strength. Okay, that's like not a bad caricature model of what's going on. Okay? Now, there are actually, believe it or not, mean field theories of randomly connected integrate and fire neurons. And the self-consistent equations for those mean field theories are a little bit complicated because you have to have a dynamical model for the temporal autocorrelation function of the current, the temporal autocorrelation of the voltage, and then you have to self-consistently solve for them. And you can see all sorts of interesting things like chaos, oscillations, asynchronous dynamics, synchronous dynamics, and so on and so forth. There's a whole kind of cottage industry looking at that. Now, this is even quite complicated because you have to keep track of spike times. Okay? There are places in the brain where spike timing is quite important. A beautiful example is the example of, like, owls are really good at this. Okay? Owls will, I mean, we're all good at this, but owls are especially good at this. Owls will hear some rustling down below. Okay? It's probably a mouse, a tasty mouse. It will turn its head and look directly at the mouse. Okay? So it's converting auditory information into a motion of the head. How is it doing that? That's called auditory localization. We can do that too, but not as well as an owl. So it turns out there's a very beautiful, simple algorithmic solution to it. So let's say this is the owl's head. Okay, this is a spherical owl. Okay, it has two ears, right? And let's say that an auditory sound occurs to the left of the owl. Okay? It turns out the owl and you also have circuitry where you have a delay line. You have one delay line coming from the left ear and another delay line coming from the right ear. Okay? Now, if the sound comes through the left ear, comes from the left, it will enter the left ear a little bit earlier than will enter the right ear. Okay? So then these two delay lines will intersect, activity propagating this way and activity propagating this way, will intersect at a particular location that depends on, the interaural time difference, the time difference between when it entered this ear versus this ear. Then you have neurons that are coincidence detectors that detect coincidences between these two delay lines. And basically the location where the coincidence detector lights up tells you where the interaural difference is. And that hooks up to circuitry that connects to the owl's neck muscles. And it basically turns the head of the owl and also the eyes of the owl directly to where the sound was. It's a beautiful, simple algorithmic solution. Does that make sense? Yeah?
You mean it's temporal correlation instead of spatial correlation?
It's converting a temporal delay into a spatial location where there's a coincidence.
Okay, then let's estimate the sound velocity is about 320 meters per second.
Yeah.
And your head is about 10 centimeters. So the time difference is much less than a millisecond level. So it's smaller than the typical neural pulse. So how
yeah, so this has to do with super resolution and having multiple neurons. So people have thought quantitatively about all this stuff. I can point you to references on that. I don't have the full numbers in my head. I actually wasn't planning on talking about this. But a lot of this was worked out by, experimentally at least, by Eric Knudsen at Stanford. He's kind of one of the big players in this thing. Okay? But I just wanted to give you an example, not to be biased, that spike timing does indeed matter for certain systems. But what happens is deeper in the brain, in higher cognitive areas that involve decision-making, executive action, and whatnot, you can do the same experiment multiple times, and a neuron will kind of fire chaotically. So the first trial, it may emit some spikes that are kind of random. On another trial, it emits some other spikes that are kind of random. And basically, the precise timing of spikes doesn't show up in any obvious way. So then it makes sense to coarse grain one step further and go from spike timing to rate. So rate, so let's do that next. So rate is just spikes per unit time, coarse grained over some time bin that you're willing to coarse grain over. So you no longer know, where, so basically you take time and you break it up into bins, and you no longer know precisely, where the spikes occur within the bin, but you know the number of spikes that occur within the bin, and firing rate is just spikes per unit time. The number of spikes in that time bin. So now if we kind of describe the output of this integrate and fire neuron in terms of firing rate, just spikes per second, it has an FI curve, a firing rate versus input curve, that's this nonlinear thresholding function. Okay? If the current is below threshold, we saw that the firing rate is 0. If the current is above threshold, it rises and rises up like that. Okay? A good exercise, this is actually a relatively straightforward exercise, is compute this analytically. This is probably just a half-page calculation. Okay? This is the origin of the nonlinear transfer functions that we see in artificial neural networks all the time. This is kind of the rate nonlinearity that shows up. Okay? And experimentalists have measured these FI curves in real neurons and so on. This was kind of was done quite a lot a while ago. So this suggests that there might be some coarse-grained dynamics just for the rate itself. And again, there's reductions that you can go through. So an even simpler model is the rate model, where basically the dynamics of the firing rate of neuron I is minus Ri plus a sum over j of all Wij of all the other neurons times this nonlinearity times the firing rate of neuron j, okay, plus any external inputs that might be coming from the outside. Okay. So this is a very popular type of model to model the coarse-grained firing rate dynamics of neurons. It turns out this is also a very powerful model in the sense that it's Turing complete. Anything that is computable by differential equations of this form for some choice of Wij. Yeah?
Does this rate have an upper limit? Like it seems this electromolecular interaction would have some kind of...
Yeah, very good question. For the integrate and fire neuron, because there's no real refractory period, you can always crank up the rate by cranking up the input. Okay? So the asymptotics, the large I asymptotics of this thing is actually linear. Okay? Real neurons do saturate. Okay? There's this kind of maximal firing rate. Okay? So this is the first thing that the integrate and fire neuron gets wrong. Okay? The real neurons kind of have a saturating nonlinearity. And that saturation comes from the the Hodgkin Huxley is hiding behind this board, but basically there's just an upper speed limit on the firing rate of the cell set by the time it takes to create an action potential. You just can't go faster than that. And the time it takes for the ion channels to recover their internal state. That sets a speed limit on these cells. The fastest cells that we see in the brain are actually these inhibitory interneurons that fire at 200 hertz. That's really fast, right? Every 5 milliseconds, they can set up a spike, okay, which is kind of twice the time one spike takes, a little over twice the time one spike takes. So there are cells that fire very, very fast. But just another basic fact, if you pick a random neuron in your brain, it'll fire about 3 hertz, even if you're doing nothing. Okay, if you're dreaming, you have these oscillations. But if you're just kind of spontaneously sitting there daydreaming, typically a random neuron will fire at 3 hertz. So there's all this spontaneous activity in your brain. Okay, so and by the way, I mean, people can look at the spontaneous activity in mice and see that it recapitulates previous experiences that they have. So they can literally see the mouse daydreaming. It's pretty amazing. Basically, when the mouse is navigating through a maze, there's a sequence of cells that fire. Then when the mouse is sleeping, that same sequence of cells fire. It's kind of amazing. Okay. So a lot of people analyze these kinds of models, but we can even an even simpler model is a spin type model, where you can move to discrete time and discretize the neurons and make them either active or inactive. And now we get to what you're used to. The spin, so each neuron is now a spin. It's just, say, plus or minus 1 or 0, 1, depending on how you we're going to use plus or minus 1, is just the spin at the next time step is just the sign. Now, because this is a more unrealistic model, we're going to go from W to J. All right. Theorists fight all the time as to whether a synapse is a W or a J. But anyways, the more unrealistic you get, the more likely it is to be J. It's a general principle in neuroscience. But and this is, you're used to this. This is zero temperature Glauber dynamics for a spin system. Okay. And there are, situations where you can get this type of reduction. Again, this paper goes very carefully. The first paper goes very carefully through the sequence of reductions.
So roughly, if the spin is 1, does it mean the rate is bigger than 10?
Yeah, exactly.
The rate is positive?
Exactly. Yeah. Yeah.
Positive or bigger than some threshold?
Bigger than some thresholds. Yeah. It depends on how you. It depends on what this nonlinearity is, and so on and so forth. But there's a mapping that goes from here to here. Okay, so this is kind of the menu of models that you have to choose from, and depending on what you want to do, you can choose one of these models. Yeah. Excellent question. We haven't talked about plasticity yet, right? So we will talk about that very soon, but there's another separation in timescales that occurs, right? Learning is usually slow and cognition is fast, right? So there is a separation of timescales where, at least over the time over which you're doing a task that you've already learned, the JIJs are fixed. But if you're learning the task, then the JIJs are changing over time. And that change is what mediates the learning. Okay? All right. Yeah.
Does it really only depend on the previous time only?
I mean, clearly not, right? So the best way to get a non-Markovian dynamical system is to integrate out degrees of freedom and pretend they don't exist, right? We've integrated out a lot of degrees of freedom, so it's definitely not going to depend on the next time step. Only on the next time step. But if you integrate out appropriately and coarse grain appropriately, then to a good approximation, the previous time step contains the most amount of information determining the next time step. Okay. And here, it's good enough. It's good enough for a government grant, right? Okay. So now let's actually do something with the simplest model. Okay, the whole business in neuroscience is connecting biophysics to cognition, right? We want to understand how the mind emerges from the brain, so to speak. So the simplest, one of the earliest examples of that was memory, right? So this is now the Hopfield model. You've actually already studied the Hopfield model in Florent's lectures in terms of a planted community detection problem. That's a special case of a one-memory Hopfield model. But let's cover it in more detail, okay? So the Hopfield model, so it's useful. We kind of take it for granted now. But it's useful to think about the conundrums that were facing neuroscientists back in the day when they tried to answer the question, what is a memory, right? A classic example of that is sort of pattern completion, right? If I show you all a picture of your grandmother, you'll be able to say your grandmother's name, okay? So you can complete partial information, right? And in fact, a lot of the experiments that are used to probe memory in animals, because animals cannot talk to us, are of this form, right? You give the animal some partial, you associate sort of, Q1 with Q2, let's say, and then or Q1 with action 1, and you present Q1 to the animal, and the animal responds with action 1 if it's learned the task appropriately, right? So this is called pattern completion. You give a corrupted pattern, and then it completes. So if we imagine that information is stored in systems of this form, where these are just spins, right, then a particular memory is a particular pattern, which I'm going to call C mu I, okay? So mu goes from 1 to p. P is the number of memories that you're storing, okay? And I goes from 1 to n. It's the number of neurons that you have in your circuit, okay? Now, these are, I'm going to call these special patterns, okay? And they contain in them the information that you want to store, such that the network can pattern complete, right? So if you present a C twiddle mu I, which is maybe a corrupted or partial version of C mu I, then the iterative dynamics of the network, which is SI, again, that dynamics over there, if I initialize that dynamics with C twiddle mu I, then that iterative dynamics will clean it up and create this cleaned up pattern C mu I. Okay? So lots of applications of this, image denoising, remembering your grandmother's name, all sorts of stuff. So the picture that you should have in your head is you have this big state space consisting of all possible spin configurations, but there are special points in the state space corresponding to these P special spin configurations. These are your memories. And there are basins of attraction that separate them. And ideally, there are no other fixed points, okay? Such that if you start with a corrupted C twiddle I, the iterative dynamics will move you to the clean thing. And if you start close to another C mu, you'll move to this one, okay? That's kind of the picture you want in your head. Okay? Now, back in the day, this picture was kind of there in verbal form, but nobody really knew how to implement it in a concrete way. Okay? Because in general, a dynamical system can do all sorts of things. It can go to a fixed point. It could oscillate. It could have chaos. Right? There's many dynamical systems that just don't look like this. Okay? So Hopfield realized that, if you have a dynamical system that has an energy function or a Lyapunov function that always decreases that as the dynamics moves forward, then you rule out oscillations and you rule out chaos. And you basically have fixed points as the asymptotic kind of state of the system. So that's what led him to this dynamic. So it turns, you all know, of course, that this dynamics has an energy function, which is just minus 1 over n, sum over ij, jij, si, sj. Okay, and you know that under that dynamics, this energy must be decreasing. Okay? So another picture, of course, is that in spin configuration space, of course, I haven't told you how to choose the JIJs in order to store these memories. Okay? But I would like to choose the JIJs so that the associated energy function has minima that are close to the desired spin patterns that I want to store in the system. Okay, this is C1I, this is CPI. Okay? So, okay, now there was another idea that was kind of flying around at the time, which was that neurons that fire together wire together. Right? This is the first time we're discussing plasticity, i.e. a rule that converts activity patterns in the network to changes in synaptic connectivity. Okay, and Donald Hebb in 1949, he had this amazing book called The Organization of Behavior. He posited this idea that maybe neurons that fire together, neurons that fire together wire together. Okay, so the basic idea is if two neurons are coactive, then the synapse between them gets stronger. Okay, so basically the learning rule, why is it that you can remember your grandmother's name and not somebody else's grandmother's name? It's because when you were growing up, you saw your grandmother and you heard her name, and so you had this coincident activity all the time, and then neurons connected together that were each elicited by the individual stimuli, the image of your grandmother and the auditory kind of experience of hearing your grandmother's name. So the idea is, while the network was learning, you initialize the spin configurations S to be one of these patterns, and then you change the synaptic weights so that it burns in the correlation between neuron I and neuron J in the desired spin configuration mu. Okay? This is literally neurons that fire together, wire together. So if these two neurons are both coactive, they're both plus, it strengthens. If one is active but the other is not, it weakens. And then this has the little biological sickness that if they're both inactive, it strengthens. But that's fine. If we did 0, 1, it would have been OK. Right? OK. So let's say we start from a tabula rasa, where the JIJs are 0. After you've seen all the patterns, the connectivity is then JIJ equals I'm going to absorb the 1 over n in the JIJ, but actually, yeah. So JIJ is 1 over n, sum from mu equals 1 to p, c mu I, c mu j. Okay? So this is the Hopfield connectivity. So Hopfield, took the ingredient of Hebb and put it into the model in this fashion, a learning rule that converts neural activity into synaptic strengths. Okay? Now the question is, this simple slogan, neurons that fire together, wire together, does it actually do anything? Does it do something useful? Does this connectivity achieve the result that I want, i.e., are the energy minima near each of these spin configurations? Okay? There's a couple of ways to see that. Okay. You can see that if you write the energy this way, then the energy function only depends on some order parameters, which are the overlaps of the spin configuration with the desired patterns. So, for example, the energy of a spin configuration, I'm going to take the 1 over n out of that definition, but the energy of a spin configuration is minus sum over ij, jij, si, sj. If I plug in the particular prescription for jij, I get sum over ij, c mu I, c mu j, si, sj. Okay? And then if I just rearrange, and I have the 1 over n now. If I just rearrange, I get 1 over n sum for mu equals 1 to p, okay, sum from I equals 1 to n, C mu I SI quantity squared. Okay, I can take the n to the inside, and then I get my extensive energy. I get sum over mu equals 1 to p, m mu squared, where m mu is the magnetization and the muth memory. This is by definition 1 over n, sum from I equals 1 to n, C mu I SI. Okay? So what's happening is that the energy is governed by p order parameters, and p very intuitive order parameters, the overlap of the spin configuration with the desired memory, right? So clearly, the energy will be low if the spin configuration aligns with one of the memories, and the energy will be high if the spin configuration is orthogonal to all the memories. Okay? So this is the first inkling that this Hebbian learning rule is doing something useful. Okay? Of course, it's just an inkling. It doesn't, there's still the issue of interference between memories, right? So in a replica calculation, so this was kind of, these are all like, the bottom 3 papers are 3 like major landmark papers, and there's really no substitute for just reading them, all right? So what I've kind of discussed is in the first paper, but in the second paper, they worked out the statistical mechanics of this model. So of course, this is a stat-mech model. I mean, you can write down the Boltzmann distribution. You can look at it in finite temperature, and what you're asking for is, are there kind of stable states of the system that are called memory retrieval states, where let's say the vector of m order parameters is, let's say, close to 1 for one of the memories and 0, close to 0 for the rest of the memories. So this is like the p-dimensional order parameter vector, right? And we're looking for thermal states or maybe 0 temperature states that look like this, okay? And what they found was they found a phase diagram. I'm just going to show you the 0 temperature version of it. Is that depending on the loading, which is the ratio of the number of memories to the number of neurons, okay, there is a threshold of loading which is about 0.14, where here you're actually okay. These retrieval states exist, all right? But beyond this threshold, they disappear and a spin glass transition sets in. And basically, there are no metastable states that have order parameters of this form. Okay? So here's the spin glass regime, and here's the memory regime. Now, interestingly, there's an earlier transition around 0.05, where actually these retrieval states are the lowest free energy. Oh, I want to turn on the light, right? It's on the left here. Okay, thanks. Oh, there, okay. I guess I kind of... All right, so beyond this regime, actually, these are the lowest free energy. Here, there are other states that correspond to mixtures or other types of states that are superpositions of multiple memories that are lower free energy. But that actually doesn't matter, because you want to use the system for pattern completion. So the basic idea is, if you give the system partial information, right, where you're kind of correlated with one of the memories, but not completely, you'll go to that memory, right? And that basically works all the way up to here, even though there are other states with lower free energy than these p retrieval states. Okay? This model is like trivial to simulate, and it's like 10 lines of MATLAB code, probably 5 lines of Python code, and just kidding. And then it's super fun to replot this order parameter and watch it evolve over time. And this is kind of a general lesson. You never want to look at neural activity itself. You want to look at the underlying order parameters or correlations between neural activity and states of the system that are of interest. The problem is in actual experimental neuroscience, we don't know what the order parameters are. That's a big problem. But, yeah? Oh, yeah, very good question. Yeah, very good question. So in your language, I would call this a planted system, where the JIJs is not IID Gaussian, but they have some correlations built into them, because the JIJs are C mu I, C mu J. Okay? However, the C mu I's, I didn't mention this, but this phase diagram is for when the C mu I's are a random IID across the mu index and the I index. So this is like random memories. Okay, so a famous fact about the Hopfield model is it's really bad at storing memories when patterns are correlated with each other. It can't, and you'll see why. I'll do a very simple calculation for why. And so something else has to happen in order for that to work. So it turns out that what the brain does, whenever it's sending information to an area that's used for memory storage, it takes the representation and expands it into a much higher dimensional representation with many, many more neurons. And that kind of whitens or orthogonalizes the representation a little bit. That may or may not help all the way. But I suspect that there's, of course, a much more intelligent learning rule that can deal with correlated patterns. We don't really know the answer to that. But that's kind of an interesting open question, is how do you store correlated patterns? There is a rule called the pseudo-inverse rule that can store any set of patterns as long as p is less than n. And it deals with that by essentially decorrelating the patterns. Okay? But the pseudo-inverse rule is not biologically plausible in the sense that it can't be implemented using local interactions between neurons and synapses, as far as I'm aware of. Okay, so now, we're not going to do this replica calculation in this course, but can we do a simple calculation that shows you why the ratio of p over n is what determines the loading, and what the mechanism is of destroying these retrieval states? And there is a simple calculation, yeah?
I was just wondering, if the strategy is to pick JIJ so that there's a minimum corresponding to each memory you want to encode,
Yeah.
then you may create fake minima which don't correspond to any memory.
That's exactly what happens here.
But do we actually think that we remember things that never happened?
Yeah, actually, there's a great book by Daniel Schachter called The 7 Sins of Memory. We hallucinate stuff all the time. This is especially a problem when it comes to like expert witnesses in court or something like that. And there's a lot of work done on that. Our memory is far from perfect. What's actually going on is much more sophisticated than this. Our brain has an internal model of how the world works. It's predicting stuff all the time. And as it gets new facts, it assimilates those facts into its internal model. So what we store is not a veridical kind of photographic snapshot of what happens. It's kind of an explanation of what we think happened. And that's what we remember. Okay? That's kind of a meta explanation for the 7 sins of memory that are kind of well documented. 7 of the sins of memory that are well documented. Yeah.
If many of the minima are just randomly created, they won't fit with our mental model, and so we'll throw them out. This is why we run all the time. There's something, like I said, there's a lot of spontaneous activity in our brain. And that spontaneous activity is thought to reflect the underlying dynamics of our mental model of how the world works. And so the actual pattern that's stored is some interaction between spontaneous endogenously created activity patterns and activity patterns that are imposed upon us by the external world. Right? So we don't understand any of that, like at a biophysics level. There's way more that we don't understand than what we do. And these types of models are just not going to cut it when we try to explain those kind of phenomena. Yeah?
This critical value of alpha basically tells, on average, we need like 7 neurons for a memory. Is that anything close to brain power?
Yeah, it's we don't really know. We don't know what P is for an adult human, right? We know some things. We know that, for example, it's actually quite sad. The vocabulary of
you know, if somebody who grew up in an educated or high-income household is about 30,000 words to 40,000 words. In a lower-income household, it's maybe 10,000 words. So anyway, that's kind of at the level of words, but that's not what P is. And we don't even know if P exists. And then we also don't know what N is. I mean, we know what N is in some brain areas, just like the hippocampus, which is responsible for storing initial memories. You know, there it's on the order of 10 to the 5th, right? So it's kind of ballpark, but these numbers are very fuzzy. Yeah?
Some memory small, then you have some use with larger weight.
That's exactly right, yeah.
And the basins?
The basins will be larger for those, exactly. Yeah. So stuff that happens to you all the time, it's easier for you to remember.
So would that be an additional weighting parameter?
Yeah, exactly. So you could have kappa mu here that up or down modulates different memories if you want. Okay, so let's do a simple calculation to see the origin of this thing. And I think I can actually do the calculation here. So let's bring this back down. Okay, so we're not going to solve the full stat mech model. Oh, let me leave this up a little bit. Okay, I'm just going to ask a very simple question. Okay. Is C mu I a fixed point of the zero temperature dynamics, of the T equals 0 dynamics? Okay. This is obviously too strong of a constraint, right? All I need is that there is some fixed point that's close to C mu I. I don't need it to be exactly at C mu I. Okay? But it's actually quite easy to analyze this strong constraint. Okay? So this is a sufficient constraint for having a retrieval state, but it's not a necessary constraint. Okay? So we can just check that by checking a certain condition. Okay? So basically, I'm just asking if is C1I equal to the sine of 1 over n, sum from mu equals 1 to p, C mu I, C mu j. Of course, I have a sum over j here. And then I have C1I. Okay, so this is, of course, my connectivity, my Hopfield connectivity. This is the initial state, and this is the final state, and I'm demanding that they be equal. Okay, that's essentially is C mu is, okay, I'm asking if C1I, the first memory, doesn't, for this random IID case, it doesn't really matter which memory I probe. Right, so I'm asking, is the first memory a fixed point of the zero temperature dynamics? Okay, it's useful to break this sum over all the memories into the special memory whose stability I'm probing and all the other memories which could muck it up. All right? So that basically boils down to breaking the sum into mu equals 1 and then the rest. Right? So if I look at the mu equals 1 term, okay, if mu is 1, then I have a C1j and a C1, oh, sorry, I screwed this up. This is a j, right? I have a C1j and a C1j here. They square to be 1, so that's gone, okay? And then I have the C1i here, okay? So, and then when I sum this over j, the 1 over n times the sum over j, again, just gives me 1. So basically, if I just restrict my attention to the mu equals 1 term, I basically get out C1i. Okay? That's fantastic. This is my signal term, right? Because I'm asking is C1i equal to the sine of C1i? And if I only had one memory in the system, you're done. You're okay. All right? However, the problem is I have more memories stored in my system. Okay? And we all experience this, right? As we remember more and more things, it becomes harder for us to remember any one thing. Okay? And it's actually known that the reason that we forget is not simply because of passive decay of our memories. It's due to our memories being overwritten or interfering from other competing memories. There's actually well-documented psychology literature on that. And you'll see this interference mathematically in the next step. So now I need to keep all the other terms from mu equals 2 to p, right? So I get 1 over n, sum over I, sum over mu equals, sum over j, sum over mu equals 2 to p. Okay. Then I get c mu I, c mu j, c1 j. Okay. So this is a lot of numbers, okay, but they're all IID random under this model, right? Everything's uncorrelated with everything else because C1j is uncorrelated with C mu j for mu equals 2 to p, right? So then by central limit theorem, the sum of all these uncorrelated numbers is a Gaussian, and I can characterize the noise just by the variance of the Gaussian. First, the mean is 0, right? Because the patterns are plus minus 1. So the critical issue is what is the variance, okay? So if we count this thing, we have a sum of p minus 1 times n terms. Okay, when p is large, what's a minus 1 between friends? We can drop it. Okay, so we have p times n terms. The variance of each term is order 1, right? So the variance of pn of them will be p times n. But I have a 1 over n here, right? So that reduces the variance by 1 over n squared. So the variance of the sum is pn over n squared, or otherwise known as alpha, which is the loading, p over n. Okay? So basically, if I summarize this thing, I'm asking the question, is C1i equal to the sine of C1i plus some noise or interference coming from all the other memories? I'm just going to call that eta I from all the other memories. Eta I is a random variable that's 0 mean and variance p over n. Okay? So the picture you should have in your head, let's just look at the special case where C1i is plus 1. So this is, of course, the local field on neuron I. And I'm just asking, is the spin configuration of neuron I consistent with the local field on it? So if we think about the local field, this is H. So this whole quantity I'm going to call HI, the local field. The distribution of the local field is centered at, say, 1, if C1i is plus 1. But there's a Gaussian distribution around it that has a standard deviation of square root of p over n. Okay? So what's clear is that as p over n becomes order 1, there's a finite probability that the local field is inconsistent with the spin configuration, so that if you initialize the spin dynamics in one of the preferred states, it will not be stable to a single spin flip. Okay? That's the outcome of this calculation. Now again, that's too strong of a constraint, because this is a recurrent iterative dynamic, so if I run the dynamics multiple steps, it might correct the single spin flip, or at least go to another spin configuration that's very, very close to this one. Okay? But the reason I did this calculation is extremely simple. It teaches you some lessons. It teaches you that when I store multiple memories in a system, when I try to recover any one memory, I can get interference between the other memories, right? And that interference gets stronger the more memories I have in the system. Okay, this is a fairly generic property of any kind of memory model that you write down. We actually had a paper called A Memory Frontier for Complex Synapses, where we proved that this exists in general for a whole range of models where the synapses themselves are dynamical systems, not just scalar values, as they are like in real brains. Number two, it kind of tells you why correlated memories are so hard to store, right? The reason this was a Gaussian of variance p over n was because this memory was uncorrelated with the rest. But if there were correlations, then this thing would become larger, right? This is why the Hopfield model is really bad at storing correlated memories. And yeah. And then now if you do the full replica calculation, you find that, like, you know, P over N is 0.14, you know, is still, the iterative dynamics can error correct. Okay? Yep.
So the phase diagram was for this dynamics?
Yeah, the phase diagram was 0 temperature, yeah. There's a second dimension to the phase diagram where you crank up the temperature and then things shift to the left. The transitions shift to the left.
Like the naive mean field equation. So what if you have like some corrections to it, like the that equation, so you shift the
No, it's fully connected. Yeah, you don't get corrections if you, I mean, they did a
like maybe a test equation and a law.
Yeah. Yeah, exactly. The phase diagram describes the behavior of the Monte Carlo dynamics, which is what we're analyzing, right?
I was wondering if you change the dynamics, you change the.
Oh, I see. Yeah. So there's a very intriguing paper by Peter Dayan and Fritz Summers that adopted a Bayesian approach to the whole thing. So what they said was, you know, we can think of the JIJs as like a conditional probability distribution given the C mu i's, right? And then we ask, what if the recall dynamics is doing is sort of trying to compute the posterior distribution of the C mu i's given the JIJs that you've stored, right? So people have played around with different dynamics. And of course, different Monte Carlo dynamics can shift the phase boundary a little bit, right? Of course, different learning rules have a much stronger effect. Like the pseudo-inverse learning rule gets you all the way up to alpha equals 1, even for correlated memories, but it's just not biologically plausible. Okay, so let's see, where are we at? Excellent. Okay, so we're already dropping stuff. But that's not bad. I got through these things. So you guys, I hear you've sort of covered the perceptron, right? Sort of. Okay. Okay, so technically I'm like 1 minute at a time, so I think this is a good place to stop. I could take some polls as to what to do next. For this lecture, you know, there were kind of, I knew I was going to cover 3 out of the 4 things really. We could do, for example, you know, next time the perceptron, or we could do unsupervised learning. So let me tell you a little bit about what unsupervised learning is, so you know what you're getting into if you want to do it. The basic idea is, you know, a lot of the learning that occurs in babies is unsupervised, right? They don't have a teacher telling them what to do. So you can kind of look out there into the natural world, and you can self-organize circuitry that efficiently processes the natural world. And so, for example, the retina. So basically, there's all these like neural response properties that we have. The retina computes spatial differences between the incoming photon stream, the center surround receptive field. V1 computes oriented edge detectors. Right? As you get higher and higher, you get these more complicated features that we don't understand. And so the question is, how is it that neural learning rules can self-organize these feature detectors just by looking at the statistical structure of the natural world? And how can you connect the statistical structure of the natural world to these feature detectors? That's what unsupervised learning is. It even goes even better. Like, your brain doesn't actually know that you have 2 eyes when you're growing up. Like, this really early on, okay? Your brain has to figure out that you have 2 eyes, all right? And it does that by detecting correlations. Correlations from neurons coming from the same eye are stronger than between eyes. Now, interestingly enough, they tested this. Well, if the brain can detect that you have 2 eyes, could it detect that you have 3? Okay, they did that experiment. They took a tadpole and they grafted on a 3rd eye onto the tadpole when it was a young tadpole. Okay? And then the brain figured out that it had 3 eyes. Okay? There's a particular wiring from the eyes to the visual cortex that occurs if you have 2 eyes. If you put in a 3rd eye, that wiring just incorporates a 3rd eye. Okay? So it's an incredible amount of plasticity. It's amazing. Of course, the story isn't always so good for frogs. They did another experiment, which was they inverted the eye. Okay? So they literally took the eye and they turned each eye upside down. And guess what this frog did when it saw a bug flying in the sky? It's sitting on a branch. There's water below. There's a sky above. It sees a bug flying up in the sky. What does it do? Jumps into the water. Okay? Your brain cannot correct for the rotation of the eyes. So these are profound issues about nature versus nurture, right? What is it about our brain that is hardwired, and what is it about it that's plastic and can self-organize in response to the structure of our world? Now, of course, we're not going to answer all of those questions, but what we're going to talk about is how neurons could do PCA. So next time we could either do perceptron or talk about how neurons do PCA. You know, I would recommend doing number 4 because it's going to help us do high-dimensional statistics later on. Okay, does that sound reasonable? Okay, excellent. So we'll start next time with 4, and then we'll go into high-dimensional statistics. All right, thanks.